Myself, Dr. Vijayanand, Associate Professor, Department of Life Sciences, Christy Jayanti College, Bangalore. So today's topic is intermediary metabolism. In intermediary metabolism, so I'm going to talk about the oxidation of fatty acid oxidation. So fatty acids undergo different types of oxidation. So we have beta oxidation, we have alpha oxidation, and we have omega oxidation. So today's lecture, we are going to talk about how fatty acids are being oxidized, especially by beta oxidation. So what is beta oxidation? So oxidation takes place at the beta carbon of the fatty acids. So that we call it as beta oxidation of fatty acids. So this pathway mainly takes place in mitochondria, and here considering the steps involved in the fatty acids. So from where do we get these fatty acids? So mainly these fatty acids are derived from triacylglycerol, which is the storage form of the fatty acids, which are mainly stored in adipose tissue. So these triglycerides are being hydrolyzed. So once it is hydrolyzed, they are converted to simpler units, especially fatty acids and glycerols. And these fatty acids, so once again, it is getting transported. So it is being transported into mitochondria, whereas short chain fatty acids, it readily diffuses through mitochondria, whereas long chain fatty acids, it require some transport molecules, we call it as carnitine. When you talk about the type of fatty acid oxidation, so there are different types of oxidation of fatty acids, so which includes beta oxidation, alpha oxidation, omega oxidation, and peroxisomal oxidation. Out of which, so most of the fatty acids will undergo mainly the beta oxidation of fatty acids that mainly takes place in mitochondria. And as a result of this oxidation, so it releases the 2-carbon unit, that is acetyl-CoA. So especially when fatty acids are being oxidized, especially in mitochondria, so there are various enzymes which are involved in fatty acid oxidation. So the enzymes which are responsible for the oxidation of fatty acids, especially the beta oxidation of fatty acids, so we'll see one by one. And first we need to understand the importance of beta oxidation of fatty acids. So the significance of beta oxidation of fatty acid, it mainly supply the energy source. And so this oxidation of beta carbon of the fatty acid, so that takes place probably in the mitochondria. So, and it results, as a result, it produces the 2-carbon unit, that is acetyl-CoA. So the first step in the oxidation of fatty acid is the activation of the fatty acids. So whether it is short chain fatty acid or long chain fatty acid, it has to undergo the activation step. So where fatty acid is being activated, so that it results with the formation of acyl-CoA. We call it as acyl-CoA or fatty acyl-CoA. So here, so the starting substrate is fatty acid. We can give example, palmitic acid is a familiar example for standard fatty acid. So when these fatty acids are being activated, so this step requires mainly ATP. So this is an energy dependent reaction. So where it is mainly catalyzed by this activation step is catalyzed by acyl-CoA synthetase or thiokinase. So ATP combines with the CoA so that it results with formation of active form of the fatty acid, that is fatty acyl-CoA. So once this fatty acyl-CoA is formed, so this reaction is mainly takes place in cytosol, that means outside the mitochondria. So as I mentioned already, so the short chain fatty acids does not require any transport molecules, whereas the long chain fatty acids, it requires the transport molecules. And especially carnitine is widely used carrier molecule, so which transfers these fatty acyl-CoA from the cytosol to the mitochondrial membrane. If you see the diagrammatic representation over here, so carnitine, so it forms complex with acyl-CoA, and this step is catalyzed by carnitine acyl transferase. This enzyme is found in outer mitochondrial membrane. So that results with formation of acyl-carnitine complex, and this acyl-carnitine complex is able to reach the inner mitochondrial membrane, so which is catalyzed by another enzyme called as translocase. Once it reaches the inner mitochondrial membrane, So this acyl carnitine, so that means converted back to carnitine. That means it regenerates the carnitine with the presence of coenzyme A. So that means it once again regenerates acyl CoA. So overall, the acyl CoA which is present in the cytosol, so it is getting transported to the inner mitochondrial membrane, so with this carnitine complex. So once the carnitine is formed, again the carnitine is reaching back the outer mitochondrial membrane, and it is ready to carry the another fatty acyl CoA, that is long chain fatty acid acyl CoA. So this transport mechanism is the major mechanism which is involved in transport of long chain fatty acids, by which most of the long chain fatty acids are being transported into the inner mitochondrial membrane. So coming to the importance or significance of this carnitine, so we need to understand the chemical structure of the carnitine. So carnitine is chemically named as trimethyl ammonium butyrate. So this trimethyl ammonium butyrate, which is synthesized mainly from lysine and methionine, and it is also synthesized from plants as well as some bacteria. And this carnitine is considered to be the transport molecule which enhance the transport of long chain fatty acids from the outer mitochondrial membrane to inner mitochondrial membrane. So this helps in transporting the long chain fatty acids into the inner mitochondrial membrane. So coming to the reactions of, that means beta oxidation of fatty acids. So the starting substrate is considered to be the fatty acyl CoA. So fatty acyl CoA, so which is the major substrate over here. So first it undergoes dehydrogenation reaction, and this dehydrogenation reaction is catalyzed by acyl CoA dehydrogenase, which requires FAD for their action. So when it undergoes dehydrogenation, so this fatty acyl CoA is converted to trans-enoyl CoA, or we call it as alpha beta unsaturated fatty acyl CoA. So this trans-enoyl CoA or alpha beta unsaturated fatty acyl CoA, again it is catalyzed by, that is hydratase. So with the specific enzyme which we call it as enoyl CoA hydratase. With the addition of water molecules, so it incorporates the OH group, probably it is converted to beta hydroxy acyl CoA. So this beta hydroxy acyl CoA, again once again it undergoes dehydrogenation, and this dehydrogenation requires NAD for their action, so that it results with formation of keto acyl CoA. So once the keto acyl CoA, because NAD is widely used as a hydrogen acceptor, so it probably accepts the hydrogen particularly at the CHOH, so that CHOH is converted to C double bond O. So once it forms beta keto acyl CoA, and this is cleaved by thiolase, which requires CoA. So in the presence of CoA, so it results with formation of acetyl CoA and acyl CoA. So otherwise we call it as fatty acyl CoA or acetyl CoA. So once this acetyl CoA is formed, then this acetyl CoA enters the TCA cycle, and it is been completely oxidized to carbon dioxide and water molecules. So to understand the reactions of beta oxidation, so these are the series of reactions by which the fatty acyl CoA so is being oxidized, and finally it is converted to acetyl CoA and acyl CoA. Talking about the energetics of the beta oxidation. So if you consider the standard fatty acid, that is palmitic acid, so when it undergoes beta oxidation, so it mainly releases 8 molecules of acetyl CoA in 7 cycles. So in each round of beta oxidation, so using the FADH2 as well as NADH plus H plus are produced, which mainly generates so 2 and 3 molecules of ATP respectively. So the total number of ATPs produced in 7 cycles of oxidation process is mainly considered as 35. In addition, we have each acetyl CoA is oxidized by TCA cycle, so 12 ATPs were generated. So in step 1, so where FAD is DH2 is involved, so 2 ATP are produced, whereas in step 3 of the beta oxidation of fatty acid, mainly when NADH plus is involved, So it produces 3 molecules of ATP. So totally it produces 5 ATPs, and for 7 cycles, so totally it is considered as 35 ATPs. So the total number of ATPs produced by the complete oxidation of palmitic acid, it includes 131 ATP molecules. So out of which 2 ATPs are mainly used for the activation of the fatty acid molecule, and the net gain is 129 ATPs. So when you calculate the energy efficiency by the beta oxidation of fatty acids, so by doing this calculation, we understand that so the beta oxidation of fatty acid, which contributes almost 40% of the oxidation. So fatty acids are predominantly oxidized by the beta oxidation process in the mitochondria. So there are other oxidation processes which includes alpha oxidation and omega oxidation, whereas when compared to the alpha oxidation and omega oxidation, so beta oxidation is the major pathway for the oxidation of almost all the fatty acids, and it contributes almost 40% of the energy. So with this, we have come to the end of beta oxidation lecture. Thank you.